Okay, so this is going to be the second part of the metabolism chapter. And now we're going to look at the actual pathways involved in metabolism. And first off, we're going to compare aerobic versus anaerobic respiration. Aerobic respiration is a series of reactions that will end up converting glucose to carbon dioxide and allows the cell to recover significant amounts of energy. The pathways involved in this are going to be glycolysis, the Krebs cycle, and the electron transport chain or respiratory chain. Under anaerobic respiration, this will utilize glucose, the Krebs cycle, and the respiratory chain, but it does not use molecular oxygen as the final electron acceptor. We're not really going to dive too much into this comparison here. We're really going to focus on aerobic respiration and also with fermentation. Now, fermentation is a form of anaerobic respiration, and it's what we consider facultative and aerotolerant anaerobes as microbes. Fermentation will only use glycolysis, and oxygen is not required for this pathway. And instead, it's actually going to use an organic compound, such as acid or an alcohol, as the final electron acceptor. And here's just an overview of the three main pathways. And sort of at the bottom there, you can see the overall net ATP gain that you might obtain. So let's start off with aerobic respiration. Aerobic respiration is a series of enzyme-catalyzed reactions. Electrons are transferred from fuel molecules such as glucose to oxygen as the final electron acceptor. The principal energy yielding scheme for aerobic heterotrophs is going to be aerobic respiration. It provides ATP and metabolic intermediates for other pathways as well. The first pathway is going to be glycolysis. Glycolysis is the starting lineup. Glucose, which is one of the main molecules that gets processed, is enzymatically converted to two pyruvic acid molecules, product of glycolysis. It may be the first phase of aerobic respiration or the primary metabolic pathway if you're talking about something like fermentation. It will synthesize small amounts of ATP anaerobically, a net gain of only 2. And pyruvic acid is an essential intermediate metabolite that is often shuttled between other pathways. So here in this figure, we're looking at a summary of glycolysis. And here you can see the enzymatic reactions that are occurring at each of the blue arrows. So glycolysis proceeds along 9 different reactions, starting with glucose and ending with pyruvic acid or pyruvate. An overview of glycolysis is presented in this figure here and contains the chemical structures and visual representations of each reactions. Each of the 9 reactions is catalyzed by a specific enzyme with a specific name. Now, we're not going to get into the individual enzymes that are involved here. The first thing that's going to happen is glucose is activated, and it's activated by adding a phosphate to it, resulting in glucose 6-phosphate. It is then converted in another reaction by another enzyme to fructose 6-phosphate. And then another phosphate is added. The resulting molecule is fructose diphosphate, meaning 2 phosphates at either end. And this is more symmetrical and can be split into 2 3-carbon molecules, as shown in step 4 of this figure. At this point, no oxidation reduction has occurred, and 2 ATPs have been used. So you're actually inputting 2 ATPs because that's where the phosphates came from, and you're actually at a net -2. The next step involves converting 1 carbon molecule, dihydroxyacetone phosphate or DHAP, into the other molecule that was produced when you split the glucose in half, which is glyceraldehyde 3-phosphate. The reason for this is that the pathway can only continue with glyceraldehyde 3-phosphate as the substrate in step 5. At the same time, 2 NADs, which are electron carriers, in the vicinity are reduced to NADHs. These NADHs will be used in the last step of catabolism within the electron transport chain to produce ATPs. Each one of these NADHs will produce the equivalent of 3 ATPs. Now, although glycolysis is the main route to pyruvic acid production for most organisms, some microbes will lack enzymes for this pathway. There are all alternate biochemical reactions that can occur, such as the pentose phosphate pathway. Our aim here is to focus mainly just on glycolysis, though. Now, pyruvic acid occupies an important position in several pathways, which is the end product of glycolysis here. And different organisms handle it in different ways. In strictly aerobic organisms and some anaerobes, pyruvic acid will then enter into the Krebs cycle for further processing and energy release, which is the pathway that we're going to see next. At the end of glycolysis, which was the oxidation of a glucose molecule, we actually yielded approximately comparatively small amounts of ATPs. On the pathway between step 6 and step 9, you end up producing 2 ATPs by substrate level phosphorylation. So for that, I'll give you a total of 4 ATPs. If you subtract the 2 ATPs that you used initially, your net gain here is 2. Don't forget also in step 5, you produce 2 NADHs. Those 2 NADHs will travel off to the electron transport chain, where they will then be processed for additional ATPs under aerobic conditions only. So as I mentioned, pyruvic acid is the central metabolite. It is utilized in several pathways by many organisms. And what we're going to do is we're actually going to continue down looking at this pathway here, where we go through the production of acetyl-CoA and then through the Krebs cycle. Now, the Krebs cycle is a carbon and energy wheel. Pyruvic acid is first converted to acetyl-CoA before it enters into the Krebs cycle. This step is not technically part of the Krebs cycle. It is an intermediate step of that cycle. And what's going to happen is you're going to go through a decarboxylation step. And we'll talk about that in a second. This oxidation reaction will release the first CO2 molecule, which is the decarboxylation of pyruvic acid. When you decarboxylate a molecule, you're removing a carbon dioxide. A cluster of enzymes in coenzyme A dehydrogenate pyruvic acid to a 2-carbon acetyl group. When you do this, when you go through this decarboxylation reaction, you are releasing electrons. NAD will be your electron carrier that picks up those electrons, and you will reduce it to NADH. NADH is formed. It will then be shuttled to the electron transport chain, where it can produce approximately 3 ATPs on that chain. So if we look at the Krebs cycle now, all reactions that occur on the Krebs cycle happen twice for each glucose molecule. And the reason for this is because we have 2 pyruvates which are formed during glycolysis. This serves to transfer the energy stored in acetyl-CoA to NAD and FAD by reducing those molecules. Remember, again, NAD and FAD are electron carriers. Those electron carriers will then pick up that energy and transfer it to the electron transport chain. The main products that you're going to observe with the Krebs cycle is that you are going to end up producing reduced NADH and FADH molecules, as well as 2 ATPs, which will be produced by substrate level phosphorylation. So here is the Krebs cycle, and we're going to kind of go through this real quick. To connect glycolysis pathway to this Krebs cycle for either aerobic or anaerobic respiration, the pyruvic acid is converted to a starting compound for that cycle. That starting compound will end up being acetyl-CoA. This is referred to as the precursor step, which is the oxidation and decarboxylation of pyruvic acid. And as you're going to end up doing is you're going to end up releasing a CO2 molecule and producing an NADH. The first step is the 2-carbon acetyl-CoA molecule will combine with oxoacetic acid, forming a 6-carbon citrate molecule. And it will also release the CoA. That is step 1. In step 2 of the cycle, the citrate will change the arrangement of the atoms to form isocitric acid. In step 3, isocitric acid is converted to a 5-carbon molecule, so we're going to lose another carbon here, molecule called alpha-ketoglutarate, which yields an NADH molecule and a CO2 molecule. Step 3 here is a decarboxylation reaction. It's actually the first decarboxylation reaction of the TCA cycle. Alpha-ketoglutarate in step 4 will then lose the second CO2 molecule and generates another NADH, plus now a 4-carbon molecule called succinyl-CoA. And step 4 is considered the second decarboxylation step of the TCA cycle. During step 5, succinyl-CoA is converted to succinic acid, and it regenerates the CoA molecule, releasing it. This releases energy that is captured into ATP by substrate-level phosphorylation. During step 6, succinic acid will lose 2 protons and 2 electrons, yielding fumaric acid, generating an FADH2 and producing fumaric acid. I always remember that the FADH comes off when you're producing fumaric acid. They both start with F. Fumaric acid then reacts with water to form malic acid. And then the final step is an additional NADH is formed when malic acid is converted to oxoacetic acid, which is the final product that can enter the cycle again by reacting with a new acetyl-CoA molecule. Now, as I mentioned before, the reactions just mentioned are for a single turn of the Krebs cycle. Each glucose will produce 2 spins of this pathway. Note that this is an enlarged, more detailed view of the middle phase depicted that we saw in the little schematic of where the 3 pathways were listed. This pathway occurs in the cytoplasm of bacteria, but it would occur in the mitochondrial matrix of eukaryotic cells. Remember that with eukaryote or with prokaryotes, they do not have the mitochondria present. Let's see if there's anything else I want to mention. So overall, the Krebs cycle serves to transfer the energy stored in acetyl-CoA to NAD and FAD by reducing them, transferring hydrogen ions to them. For that reason, the main products of the Krebs cycle are these reduced molecules, as well as the 2 ATPs for each glucose molecule produced by substrate-level phosphorylation in step number 4 to 5. The reduced coenzymes NADH and FADH are vital to the energy production that will occur in the electron transport. Along the way, the 2-carbon acetyl-CoA joins with a 4-carbon compound oxoacetic acid and then participates in 7 additional chemical transformations while spinning off NADHs and FADHs. That is why we call the Krebs cycle the carbon and energy wheel. Now, the respiratory chain, this is also referred to as the electron transport system. It is a chain of special redox carriers that receives electrons from NADH and FADH. Electrons are passed sequentially from one redox molecule to the next. The flow of electrons allows the active transport of hydrogens outside the cell membrane. And oxygen receives the hydrogens and electrons and will end up producing water. So in this figure here, we're looking at the electron transport system and oxidative phosphorylation, which is the process that occurs at the electron transport system. And this is occurring in bacterial membranes. The only membrane in bacteria or prokaryotic cells, remember, is its phospholipid bilayer. Starting at NADH dehydrogenase, electrons brought in from the Krebs cycle by NADH are passed along the chain of electron transport carriers. Each adjacent pair of transport molecules undergoes a redox reaction coupled to the transport of electrons is simultaneously active transport of protons into the periplasm by specific carriers that are present. These processes set the scene for ATP synthase and the final proton and electron acceptance. You do want to note that you want to note the differences in this figure here of the final electron acceptors that are occurring in aerobic versus anaerobic respiration. And not that you necessarily need to know this, but the sequence of electron carriers in the respiratory chain in most aerobic organisms is NADH dehydrogenase, that's the first one, followed by flavin mononucleotide, FMN, then coenzyme Q, then cytochrome B, C1, cytochrome C, and cytochromes A and A3, which are complex together. Now, the electron transport system is on the inner mitochondrial membrane of the eukaryotic cells. The electron transport system on the inner mitochondrial membrane Cristae. As the carriers of the mitochondrial Cristae transport electrons, they also actively pump protons to the intermembrane space, producing a chemical and charge gradient between the outer and inner mitochondrial compartments, as shown in this figure here. Now, ATP synthase, this is stationed along the membrane in a close association with the electron transport system. ATP synthase will capture released energy from the electron transport system carriers. Oxidative phosphorylation is the coupling of ATP synthase and to electron transport. Each NADH that enters into the electron transport system will give rise to approximately 3 ATP molecules. NAD and FMN enter the electron transport chains at different points, so there is less energy that is given to FADH molecules and will give rise to approximately 2 of those. The process of linking the electron transport system with ATP synthase is called chemiosmosis. As electron transport carriers shuttle electrons, they actively pump hydrogen ions or protons into the periplasmic space, or the space between the wall and the cytoplasmic membrane. Depending on whether the bacterium is gram-positive or gram-negative, this process sets up a concentration gradient of hydrogen ions called proton motor force. The proton motor force consists of differences in charge between the outside of the membrane, positive, and the inside of the membrane, negative. Separating the charge has the effect of a battery, which can temporarily store potential energy. This charge is maintained by the fact that proton can't cross the membrane. The only site where protons can diffuse into the cytoplasm is the ATP synthase complex, which sets the stage for the final processing of protons, leading to ATP synthesis. ATP synthase is a complex enzyme composed of 2 large units. It is embedded in the membrane, and part of it rotates like a motor and traps chemical energy. As the proton ions flow through the center of the enzyme by diffusion, the other compartments pull in ADP and phosphate groups. Rotation causes a 3-dimensional change in the enzyme that bonds the 2 molecules. thereby releasing ATP into the cytoplasm. The enzyme is then rotated back and start position and will continue the process. Eukaryotic ATP synthesis occurs by means of the same overall process. However, eukaryotes have an ETS station in the mitochondrial membrane between the inner and outer mitochondrial membranes. So finally, or just to recap, chemiosmosis, as the electron transport carrier shuttle the electrons, hydrogen ions are actively pumped into the periplasmic space, or the space in the cell wall and the cytoplasmic membrane. This sets up a concentration gradient of hydrogen ions called the proton motor force. This proton motor force consists of the differences in charge between the outside and the membrane, outside of the membrane, which is positive, and the inside, which is negative. Separation of charges temporarily stores potential energy. The protons can only diffuse into the membrane through ATP synthase, which sets the stage for ATP synthesis. ATP synthesis in eukaryotes occurs through the same overall process, except again, it's occurring on the inner mitochondrial membrane. Now, the overall theoretical yields for aerobic respiration. To attain theoretical maximum yields of ATP, we assume a ratio of 3 for the oxidation of NADH and 2 for FADH. The actual yield is generally lower and varies between eukaryotes and bacteria among bacterial species. These are just, again, overall theoretical yields that you can produce. So the overall summary of aerobic respiration is that you are going to end up with a possible production of ATP that is 40, 4 from glycolysis, 2 from the Krebs cycle, and that was by substrate level phosphorylation, and 34 from the electron transport. However, because 2 ATPs were already spent in early glycolysis, this leaves us with a maximum of 38 ATPs. The actual totals may be lower in certain eukaryotic cells because energy is spent in transporting NADH produced during glycolysis across the mitochondrial membrane. Certain aerobic bacteria come closest to achieving the full 38 ATPs because they lack mitochondria and, for that reason, do not have to use ATP and transport of NADH across the outer mitochondrial membrane. In addition, you are producing 6 carbon dioxide molecules, which are generated during the Krebs cycle, 4 actually on the cycle, and 2 in the precursor step. You're also going to have 6 oxygen molecules are consumed during the electron transport, and 6 water molecules are produced in the electron transport and 2 in glycolysis. But because 2 are used in the Krebs cycle, then this leaves a net gain of 6. So the terminal step, this is catalyzed by the enzyme cytochrome AA3, also called cytochrome oxidase. It is adapted to receive electrons from cytochrome C, pick up hydrogens from solution, and react with oxygen to form water. And that is essentially how your water is being produced on this. Okay. So anaerobic respiration, nitrate and nitrite reduction systems. These are typically found in E. coli. Nitrate reductase catalyzes the removal of oxygen from nitrate, reducing it to nitrite and water. A test for this reaction is one of the physiological tests that's used in identifying E. coli. Denitrification, enzymes that further reduce nitrite to nitric oxide or nitrous oxide, and even nitrogen gas. Other organisms can actually take this to those steps. This is also important in identifying those bacteria. Now, let's shift gears a little bit here, and let's now talk about fermentation. Fermentation is the incomplete oxidation of glucose or other carbohydrates in the absence of oxygen. It uses organic compounds, such as acids or alcohols, as the terminal electron acceptors. And this is going to end up yielding much more smaller amounts of ATP. So again, the definition of fermentation is the incomplete oxidation of glucose or other carbohydrates in the absence of oxygen. Overall, the term fermentation has acquired several looser connotations. Originally, Pasteur called The microbial action of yeast during wine production ferments, and to this day, biochemists use the term in reference to the production of ethyl alcohol, but yeast acting on glucose and other carbohydrates. Fermentation is also what bacteriologists call the formation of acid, gas, and other products by the action of various bacteria on pyruvic acid. The process is a common metabolic strategy among bacteria. Industrial processes that produce chemicals on massive scale through the actions of microbes are also called fermentations. Each of these uses is acceptable for one application to another. So the overall chemistry of the fermentation systems that produce acid or alcohol. In both cases, the final electron acceptor is an organic compound. Acids and alcohols are organic compounds. In yeast, pyruvic acid is decarboxylated to acetaldehyde, and the NADH is given off in the glycolytic pathway, reduces acetaldehyde to ethyl alcohol. In a homolactic fermentative bacteria, pyruvic acid is reduced by NADH to lactic acid. Both systems regenerate the NAD to feed back into glycolysis or other cycles, releasing the electrons off of the electron carriers. So here's just two examples where you're looking at two different pathways. And again, the pathways that exist are really going to depend on which enzymes are present there. Now, the products of alcohol fermentation, this occurs in yeast and bacterial species that have metabolic pathways for converting pyruvic acid to ethanol. The decarboxylation of pyruvic acid to acetaldehyde, and then the reduction of acetaldehyde to ethanol. NADH is formed during glycolysis, is oxidized, regenerating the NAD and allowing glycolysis to continue, keeping the cell alive because metabolism still is continuing. Now, products of acidic fermentation, these pathways are extremely varied. Homolactic fermentation is lactic acid bacteria reduce pyruvic acid to lactic acid mainly. Heterolactic acid fermentation, glucose is fermented to a mixture of lactic acid, acetic acid, and carbon dioxide. Members of the family Enterobacteria possess enzyme systems for converting pyruvic acid to several acids simultaneously. And in fact, they can actually produce acetic acid, lactic acid, succinic acid, formic acids, as well as CO2. This fermentative activity accounts for the accumulation of some types of gas in the intestines. Some bacteria will reduce organic acids to a neutral 2,3-butadiol and other solvents. Okay, now just shifting gears a little bit, because glucose is not the only molecule that exists, we can have other molecules that can be broken down for energy. And that would include lipids. Lipases will break apart fatty acids joined to glycerol. The glycerol will be converted to dihydroxyacetone phosphate, which is DHAP, and that can actually end up entering back into step 4, glycolysis. Beta oxidation is a process that occurs of fatty acids. 2 carbon units are transferred to coenzyme A, creating acetyl-CoA. And once you create the acetyl-CoA, you then can go and enter into the Krebs cycle. Oxidation of 6-carbon fatty acid molecule can yield excess of 50 ATPs compared to 38 for 6-carbon sugar. So you can end up producing a lot more energy by breaking down fats than you can by breaking down sugars, which is why cells oftentimes will use fatty acids as the main store for energy. Proteins can also be broken down. Although proteins are typically just broken down for amino acid pools, proteases will break down proteins to their amino acid components. Amino groups, which are going to be removed by a process called deamination, must occur. And the remaining carbon compound can easily be converted to one of several Krebs cycle intermediates. Now, the frugality of the cell. Amphibolic is most catabolic pathways contain strategic molecular intermediates that can be diverted into anabolic pathways. A given molecule can serve multiple purposes to derive maximum benefit from all nutrients and metabolites. Catabolic and anabolic pathways are integrated to improve cell efficiency. And here's an amphibolic view of metabolism, where you can have these intermediates really work between several different pathways. Amphibolic sources of cellular building blocks. The precursor molecule is a compound that is a source of another compound. For example, glyceraldehyde 3-phosphate can be diverted from glycolysis and converted into precursors for amino acids, carbohydrates, and triglycerides. Pyruvate as a precursor can provide intermediates for amino acids. Gluconeogenesis is when you take pyruvate as the starting point for glucose synthesis in the event of inadequate glucose supply. Acetyl-CoA can act as a precursor and can be converted into one of several amino acids. It can also be condensed into hydrocarbon chains for fatty acid and lipid synthesis. Precursors to DNA and RNA, pathways that synthesize purines and pyrimidines originate in amino acids, and they can be dependent on intermediates from the Krebs cycle. During times of carbohydrate deprivation, organisms can convert amino acids to intermediates of the Krebs cycle and derive energy from proteins. Crucial role of glucose in metabolism and energy utilization. And sometimes it's important that we be able to synthesize carbohydrates. The biosynthesis ensured by several alternate pathways. Major components of cellulose walls and storage granules depend upon this. Glucose 6-phosphate is used to form glycogen. Proteins. Proteins are a large proportion of the cell's contents. It is essential components of all enzymes, cytoplasmic membranes, the cell wall, and cell appendages. There are 20 amino acids that are needed to make these proteins. Some organisms, such as E. coli, have pathways that can synthesize all 20 amino acids. Cell divisions. If you think about cell division, as far as metabolism is concerned, 2 cells need twice as many ribosomes, enzymes, etc. The cell wall and membrane nearly double. Phospholipid bilayer components assemble spontaneously with no energy input. Proteins and other components must be added to the membranes, and the cell wall growth requires energy input. Finally, the third way to make ATP is photosynthesis. And photosynthesis is essentially taking the process of light energy and converting it into chemical energy. I do not expect you guys to know the pathways of photosynthesis, but they are up here. If you would like to rehash things you may have learned in biology. And that is it for chapter 8.